So like I said, my name's Eva Brayfindley. I'm a data scientist here at PNNL. I do a lot of work doing data science and machine learning for chemistry applications. Specifically, I work in metabolomics. And so I'll be talking to you a bit about the data science workflow. In order to do that, I first want to start off with some definitions. So we'll start off talking about what data science is, what artificial intelligence is, and what machine learning is. We'll talk through some of the differences and the sorts of things we can do with that. And then we'll talk through the data science project workflow and what all is involved in that, and some important principles to think of while you work through data science type projects. So to start off with, oh, someone else hitting slides because I have not clicked anything. Okay. All right, so to start off with, we're going to go with some definitions. What is artificial intelligence and what is machine learning? Typically, we think of artificial intelligence as being kind of this very, very broad umbrella term. It's basically anytime you create a machine to mimic human behavior. It can be very, very simple stuff. So if you've ever called up someone on the some company on the phone and got their kind of automated menu of press 1 if you want to talk to someone, that would actually be considered artificial intelligence. But we don't think of that as machine learning. Machine learning is a subset of AI, and it very specifically builds models based on some data that it gets fed its training data to make predictions. Deep learning is another thread that you will hear, another term you'll hear a lot, and that's a very specific subset of machine learning, a very specific class of algorithms and models that get used for highly complex problems. Data science, on the other hand, is a little bit, there's a little bit of overlap, and then it's also a little bit distinct from all of these things. So that Venn diagram is a little bit off to the side, not quite within these umbrella terms. There are parts of data science that are a subset of AI, but data science also includes lots of statistics, scientific methods, and things like that, with the overall goal to extract meaning and insights from data. So that's all well and good from a definition point of view, but what does that actually mean practically? And what are the sorts of things you'll do if you're doing data science versus machine learning? So when we look at these Venn diagrams again and break it out into the sorts of tasks that we do, data science encompasses everything from data preparation, statistics, visualization, and also includes some of the model building that you'll see in machine learning, like decision trees and Bayesian methods, things like that. So it's this big box that kind of covers everything. Machine learning and artificial intelligence are more specific. They focus on the model building portion of things and less on some of the other analysis work that's happening. Now, this is just my definition. This is not necessarily everyone's definition and breakdown of how they separate these categories, but this is what I think of when I think of data science, machine learning, and AI. Okay, so now that we know what data science is, we'll talk through the project workflow, and this will include the project workflow for both machine learning specifically and for data science generally. So we'll get into all of these. Okay. So this is more or less what the data science workflow looks like. Now, I'll caveat that by saying I haven't put anything about deployment here. So this is up through building your models and understanding your data. If you do want to put this into play in the real world, there's some extra steps past this to think about. But here's how we kind of define and think about our data science workflow. We have a couple categories of what we think about. So you'll see the data exploration, the model development up there, but it kind of breaks out into these boxes. You start off by defining your task for machine learning or for data science. Are you going to analyze your data? Are you going to classify your data? Are you going to try to fit a line to your data? And that's the sort of task definition you start with. You can then move into selecting and describing your data. So it's incredibly important to identify data that's relevant to the tasks that you want to do. And there's a couple ways you can describe data that we'll get into in a few minutes. That data then has to be prepared to actually get used by a computer for this sorts of analysis. So there's all kinds of pre-processing to be done and cleaning. Things like making sure there are no zeros or missing data in your data set, that everything is labeled, everything's normalized, set up right, that the dates aren't swapped in your data set. From there, that data gets transformed into something that's highly useful for either data science or machine learning. This is not always a step that's included, but is frequently included in these workflows. And what we do in this step are things like dimension reduction. So if you have two features that are very, very similar, maybe one of them winds up being actually useful for your model building, and so you can get rid of the other or not use it so much. And that's what we talk about with dimension reduction. We then do some very involved data exploration. This can be visualization, it can be statistics, it can be data mining, and kind of partnered with this and down below it is the machine learning. Included in the exploration can be some model building, or it can be a slightly separate task. You can do some model selection, some building, some training, some evaluation, kind of build that machine learning as either part of your data exploration or in addition to your data exploration. And finally, where you land at the end of this is with your interpretation and evaluation of the results that you've done so far. This includes uncertainty estimation, statistics, talking with subject matter experts. Overall, this workflow has a few different categories, and we'll get into each of these more in depth in a few minutes. But I want to point out that I have put arrows one direction on this workflow, and that's not actually true. Each one of these steps can influence the step before it or the 5 steps before it. So it's more of a circular process than it's shown on this slide here. But it's useful to think about it broken down into these tasks. And as you start to approach these problems, make sure you are breaking them down into these kind of buckets and you know that you're checking the boxes on all these steps. Model development is also included in the data exploration. And you can think about model development as being a way that you explore the data. And that's why I break it out that way. So this is the workflow we'll be going through. The machine learning model workflow is that same workflow, but I'm going to break it up a little bit differently so that we can talk through each of these. So in this case, we're going to talk about task definition, just like we did a second ago, the data collection, the modeling and training, so that machine learning bucket down below, and then the evaluation at the end. Like I said, each of these are very, very related, and it doesn't just go one direction. You don't start with a task, end with evaluation every time. Sometimes you know what your data is and you define your task based on that data. Sometimes you get to the evaluation stage and your model hasn't done very well. So you start over completely, right? So it's very organic, very, very circular, it goes back and forth. So the very first step is your task definition. Identifying the task is absolutely critical. It's really not a great idea to just throw data at a model without understanding what you're trying to accomplish. It's useful to start with identifying the task and thinking about what data or modeling might be necessary to actually achieve a successful workflow. You really need to start off thinking about what you're doing before you can actually do it. So to that end, what are some of the tasks that we do with data science? What are some of the tasks we do with machine learning? Now with data science, a lot of it is more focused on the data itself. With machine learning, a lot of it is more focused on the model or the task that we do with that data. So for the data science side of things, you can collect the data, you can store the data, create a database, do something like that. There's lots of data cleaning and exploration involved on the data science side of the house. Data integration, so taking multiple streams together and merging them, characterizing what's present, what's not in a data set, what that data set looks like, visualizing all those characterizations. All of these things are the sorts of tasks we do with data science. A little bit different is what we do with machine learning. To me, again, this is a very personal definition. There's probably at least a few people in the audience who would disagree on some of my breakdowns, but what I think of when I think machine learning is more that task oriented. What's the model going to do? And in this case, and this is kind of how I've laid it out. So what we do with machine learning often is more classification or regression or forecasting, so predicting something. And when you get into the more involved kind of deep learning things, you can also get into things as crazy as generative art or all the chat GPT that you've seen in the news, stuff like that. So you can also get very big into these very large models with machine learning. So there's typically two types of machine learning that we think of. There's supervised and there's unsupervised machine learning. The supervised side of the house, the goal is to learn to predict a particular outcome. So you have inputs, you have known outputs for a certain subset of that data, and you try to learn the relationship between those inputs and outputs. It does require you to have that labeled data, at least during the training phase. And it also requires that the data be super representative of the relationship you're trying to learn. Basically, with supervised data, you've got the answer key and you've got the test, and you're trying to learn how to get that answer key from the test. Some pretty common tasks when it comes to this are things like classification. Is this picture a dog or a cat? Or regression, draw a line or some kind of curve through your data set. Now, on the other side of the house, we have unsupervised learning. And the goal from unsupervised machine learning is to understand the data. The tasks look a bit different when it comes to unsupervised because we do not have any labels. So what we're really trying to do is just understand some of the patterns in the data set as they are without any labels or anything attached to it. One example that you'll see a lot in biology that you may or may not know is unsupervised machine learning is principal component analysis. So this is an unsupervised method where PCA is applied to basically highlight the variance in your data set and reduce the dimensions down to your features that have the most variance. Common tasks in unsupervised learning include clustering and anomaly detection, and some other more involved things like autoencoders that we are not going to get through today. So this is when it's worth, this is why it's important to define the task. You don't know if you're supervised or unsupervised unless you know what you're trying to do and you know your data that you have, right? So in the kind of top left of this whole graphic, we have an example for regression where you have weight and height on your axes and you're trying to build a model that predicts weight as a function of height. So you've got your two features trying to build a model between them. Now, on the other end of the spectrum, you've got the rather complex machine learning that you see down on the bottom right. And I'm actually going to do A in the roughly the middle of the screen here. So in this one, you've got a multi-layer perceptron model, which is also known as a neural network. And the goal of this particular model is to actually predict toxicity from molecular properties. So this is a very, very different kind of problem. You're actually predicting features based on the inputs you give it. And those inputs look very, very different than they do in the example where you've got regression, weight, and height. So knowing your task, knowing what you're after, that helps define the models and the approaches that you can take. So if there's nothing else you take away from this portion, think very, very hard when you first start doing data science about the task definition that you have. Make sure that you are actually doing the step, don't skip it. Having a problem to solve is not the same as framing that problem for data science. And it's very important to understand that distinction. Always talk to your subject matter experts as you work through your problem. Don't work in a vacuum because you might have the problem the wrong way around, or maybe be answering a different problem than you think you are. So always talk with people, make sure you understand what you're up to. And then it's very important to understand that data can also impact your task definition. If you want to do supervised classification, but you don't have labels, you can't really do that. So that's the sort of thing you need to think about as you define your task. Speaking of, data collection is an integral part of the work that we do as data scientists. We are humans using human design tools to collect data. So what that means is the data is not always perfect. Exploring and analyzing that data is incredibly important to understand if that data is actually relevant to the task that we've defined, and then to identify or mitigate any biases that might be in that data. So I put this graphic back up on the screen this time because these are all the data exploration steps. You need to start off by identifying relevant data, describing that data, understanding what's there, how much, all the formats and representations that that data is in or might need to be in order to be able to do machine learning or data science. Clean and pre-process that data however you need to, and then explore that data to identify any initial trends that may impact your model selection. So what is data? Traditionally, data comes from experimentation, and it still does when you're talking about biology and talking about chemistry and things like that. You still have a lab notebook where you're collecting experiments, you're writing down all the results from your experiments in the lab. But on the other hand, you also have all sorts of other things coming through now. So things like social media or the smartwatch that you wear and all the biological data, all the kind of human response data that collects. Everything is data, and it's really important to understand the format of your data so that you know how to use it. And the way you understand that data is by describing the data. Now, I'm not going to go through each of these in depth, and you can look at some of the 5 V's of big data later in more depth. But the way that you describe your data typically comes down to what's called these 5 V's of data. You have the volume, which is the amount of data that comes through, the variety, so the type of data. Is it free form? Is it structured? Does it come in a table? Does it come in text? Does it come in images? Velocity is the speed at which that data is generated. So is it going to be streaming terabytes of data to you every single day, or is it an image that gets captured once a month or something like that? Veracity is the degree to which data can be trusted. Now, when you use the data from your own labs and things like that, that's maybe less relevant, but veracity is incredibly important when you think about open source databases, right? So you want to use like NIST for their GCMS libraries, and maybe not some spectrum you get that doesn't even have an author, right? So those are that's something to think about when you're looking at open source. Value is the business value of the data that's collected. That can start to impact things like the security that you need to keep of the data. So this is how we describe our data. However, the real place to start is, does the data answer my question? Does it work for the task that I've defined? And there's kind of 3 things that you think about when you start to try to answer, does it answer my question? So the first thing is, why was this data collected? Data collected for one purpose may not actually be sufficient or complete for another purpose. So do be aware of that. The one example here is that data that verifies engineering specifications, that something is up to spec or matches with quality assurance, may not actually predict its performance in the field. Those are 2 very different data sets. And that can be kind of hard to understand or kind of identify those differences. The second big question is, does the data sample the entire space of interest? When you're talking about biology and chemistry, in the world I work in with small molecule metabolomics, the chemical universe is absolutely enormous, right? Just so huge. And we can't possibly have experimental data for everything that we could possibly want to look at. But do we at least have data that focuses on the region of interest? Some of the examples for this are the curated databases that you'll see out there. So things like the Human Metabolome Database or Drug Bank. They focus on very specific subsets of the chemical universe. So if you're going to use those databases, make sure that that's actually the space of interest to you. If you're using Drug Bank, which focuses on drug-like compounds, but you're not actually looking at drugs, it may not be the most useful data set for you. And then the last one is whether the training environment actually looks like your deployed environment. So if you go to put this model that you've built into the real world, is it going to be looking at the same sorts of data that you've trained this model on? And that's very important to think about. Okay. The next step goes into some of the pre-processing, but before we can talk about the pre-processing, we need to talk about data formats and representations. There are lots of possible data formats, and you have to choose something, right? And the goal is to choose the best format that you can for the task that you're doing. Each representation, and I'm talking things like images, text, numbers, graphs, whatever, has different strengths, and the ideal representation may not be obvious for you. So what it's good to do is go out into the literature, see what's being done, talk to some other people who've worked in this space before, see what they've worked with, and see if that will work for you. One example of this is molecular structure representation. So you can write the things like a molecular formula down. You can also write down like a smile string, which has a little bit more structural information on it. You can draw your kind of ball and stick model and take a picture of that, for example, as your input. But each representation is basically a different way to represent structural information. And each of those representations may even require a different model type. So it's important to know, okay, am I going to take images of ball and stick models? All right, I need to use a model that's going to work with images. Am I going to use text? So am I going to use a smile string for everything? Okay, well then I want to use something that's good for text modeling. So it's important to understand those and what you need to be using depending on what you've got. You can also do data preparation to transform your data into the format that you want to use. So if you have a mix of molecular formulas and smile strings, do you want to maybe convert everything to smile strings first, use something like that. So there are some common standards for common data formats. I just showed you some of the molecular representation formats available to you. And all of the ones listed there are fairly standard at this point. There's strings and graphs, and you can make a table of your feature sets and all sorts of other things that you can do to represent any kind of compound. When you talk about experimental data and data that comes off of an instrument in the lab, you will also get into instrument-specific formats. And those can be a little challenging to deal with. There are some instruments that have kind of their manufacturer-specific format, right? And if you're collecting data from different instruments, maybe you need to convert everything to one format first. So that would be, step one in your data preparation. The other thing to be aware of when you're doing pulling information off of an instrument is that it may already include some calibration that instrument has been done. So some default thresholding or something like that, or it may not. And that is something that you have to be aware of when you're using that data. One very, very key part to this is that an experimentalist and a data scientist may not do the same sort of pre-processing. They may not need the same sort of pre-processing, and it can actually be somewhat detrimental to each other to have the same sort of pre-processing. So it's always good to have the raw data and then pre-process as needed. So as an example of this, when you're looking at a mass spectrum, it can be really, really good to have thresholds for noise removal. So you're not seeing a bunch of peaks with an intensity of like 2. However, for machine learning, if you get rid of all that noise, it can actually make the model that you build really, really brittle to noise in future. So it won't be able to actually deal with noise that comes up in the future. And so that's just one kind of simple example of how those differ for a data scientist and experimentalist. The thing that you will hear from every data scientist ever for the rest of time is that a model is only as good as your data. So go about your data selection and your data prep and your data transformations very, very carefully. Be very thoughtful in what you're doing and the work that you do. Make sure you talk to subject matter experts for insight into key features that might be present in that data. Select a format appropriate for what you're doing. And then make sure that you're exploring the data properties so that you can understand what that data looks like so that you can succeed in your workflow. All right. Next up is modeling and training. This one is kind of specific to the machine learning because we're building machine learning models here, but you don't have to. You can start with statistical models and things like that. The goal is to start simple and move to more complex architectures as you need. And at this point in kind of the machine learning literature, there are baseline models and methods that exist for the majority of problem formulations. So start with them before you develop more complex models. There's some kind of existing standards for things like image classification. So start off with those kind of standard models and then tailor as needed. When you do this model selection, it's really important to consider what your data looks like. So your inputs, do you have an image? Do you have text? Your output goals, do you want a classification or do you want a prediction? What some of the features of the data and the quality of the data that the model might have to deal with or use. And then of course, like I mentioned, the literature norms for all those other features. That's not to say that you shouldn't start from scratch and try something new. But if you do run into a wall, if you have some issues knowing where to start, the literature can be a great place, and there's lots of existing models you can play with. One of the other things that you'll get into is model training parameters. So how you train that model, how you transfer it to your task, things like that. And those are going to vary with the models that you look at. They can also be fine-tuned absolutely forever. You can always go from 99.6% accuracy to 99.7, but it may not be worth it to do that until you really need to. So start with some defaults until you get a better idea of what you need and the level that you need to do it to. When we talk about model selection, it's not just a function of the available data, the quantity and quality, but the model that you want to use is also has some inherent capacity. So things like linear regression, where you try to draw a line through some data, is a lot less complex than some of the big deep learning things like you see in the news, like ChatGPT, right? So when you're talking about linear regression, you have a line, you have the slope and the intercept, and those are kind of your parameters. You have a X and Y, and that's it. When you talk about things like GPT, you have billions of parameters that your model can learn. Don't use GPT when linear regression will suffice, and don't use linear regression when you need GPT. So understand the limitations of the models that you have and what you might need in future. So this is a workflow that I like. It comes from a paper in Nature that's machine learning for biologists. And this is a pretty good workflow for how to do some of your model selection. So the model you select, like I said, is about the data that you have, quantity, quality, and then also the complexity of the model. So when you get to the point of selecting a model, step one, do you have sufficient data? Yes, no. And then you can move through this workflow to understand more of what you can do with it. So are you predicting values or class? If you're predicting a class and have labeled data, maybe you could do that support vector machine. And that's just straight down that kind of workflow. So this can be a really handy way to start off with how to make decisions for your model selection. And then in that same paper, so that's the machine learning for biologists in Nature, there's also some common models for biological problems. So you've got some examples here for mass spectrometry or images or molecular structure, protein sequence, and some recommended models and challenges that you might have when you do that. So what are some common pitfalls that you might run into? One of the biggest ones is that you build a model that's measuring the wrong thing or working with the wrong data, or accidentally using like the row name rather than the data itself, something like that. So be careful of things like that. There's kind of that age-old phrase that correlation is not causation, right? So make sure that you're still sticking with that when you do this kind of data science and machine learning approach. It can be easy to forget that when you're doing these kind of larger models. There's also overfitting and underfitting, which are both big problems when it comes to machine learning. Overfitting is when you fit a model so precisely to the training data that it can't possibly look at new data because it's only ever going to do exactly what it did on its training data. It just can't take in any new data and do anything well. Underfitting is cases like when you draw a line on something that should be a curve, right? So you're just totally underestimating the complexity of the data that you have. And both of those are problematic. And then finally, the last big one is losing sight of the objective. Keep your task in mind, make sure that you're still working towards that task definition that you have. So to talk about underfitting and overfitting, I've got some examples down at the bottom of the data points, what underfitting looks like and what overfitting looks like. Fitting your training data perfectly, if you get 100% accuracy, you should be a little bit suspicious. A perfect score is a little bit scary. Your model may be so overfit that it does perfectly on your training, and then you try to use it in the real world and it falls apart completely. But then again, on the other side, don't not train your model at all. Don't draw a straight line when it should be a curve. So it's kind of this sweet spot that you're trying to hit. You want to get it just right. And that's very hard and very challenging. So work with some experts, play around with your models, look at the literature and how people deal with these to understand better how you can solve them. So for some of the key takeaways for training, you do need to be careful when selecting your model. You want to make sure that you have both enough and the right type of data for the model that you choose. You can look in the literature, kind of that table that I showed very briefly, and check what other people have done to see if it's going to work for you or if you need something new or different. And then, of course, always talk to your SMEs, your subject matter experts, and other data scientists if you're not sure where to start. Be careful with overfitting and underfitting. It's very hard to get it just right. It's kind of that Goldilocks fit. But if you can, get as close to that just right as you can. That's the goal. Okay, and the last step in the workflow that we're going to talk through is evaluation. You typically, when you're doing evaluation of the model and the work that you've done so far, you want to have both quantitative and qualitative results. So a graph that looks nice, and also the kind of numbers that go along with that, right? You want to ensure that your model output is unbiased and fair. And just as a reminder, if you get to the end of this process and you do the evaluation and it turns out your model absolutely sucks, you are totally allowed to go back in the process and try again at the modeling and training stage, or even all the way back to the task definition. Do you need to change your task a little bit so that you can actually address it with what you're doing? So metrics are the quantitative part of your evaluation, and they depend almost entirely on the problem formulation and what you're after. There are metrics that are really useful to the model, metrics that are really useful to the people reading the results, and metrics that solve both, right? And so you want to be aware of what you're selecting and what you're using for your evaluations. Metrics can also be very misleading depending on your data or your problem formulation. So I'm going to start off with a few metrics that we see a lot when it comes to kind of biological and chemical classification problems. But this is, again, very specific to classification in particular, so one sort of task. There are other metrics for other tasks. But a lot of times what you'll see are true positives, false positives, true negatives, and false negatives. I'll define those in a second. A confusion matrix is shown down at the bottom. This is just one representation of showing some of these numbers, and you'll see this a lot when you look at machine learning papers. You'll see confusion matrices come up. The goal is a really high true positive and a high true negative, and then a low false positive and false negative. So those blue boxes, you want to be high, and the yellow boxes, you want to be low. Okay, so what is a true positive? A true positive is if you predict the thing, and that's the real label for that thing, right? So if you have a picture of a dog and you put it in your model and it tells you it's a dog, that's a true positive. If you have a picture of a cat and you put it in your dog predicting model, if the image is the cat, but your model predicts a dog, that would be a false positive, right? Now, if it's a cat and your model predicts it as a cat, then that's a true negative. But if it's a dog and your model predicts it as a cat, that would be a false negative. Okay, so we've got all these numbers, right? You've got false positive, true positive, false negative, true negative. What does that actually mean, and how do we actually use it? So typically there are two sorts of metrics that you'll see in the literature outside of your kind of standard accuracy label. And these are precision and recall. So I'm just going to show these to you very quickly. Precision is a measure of how many selected items are actually relevant. So in this case, the things that are predicted as true positives divided by all the things predicted as positives. So the number of images labeled dog relative to accurately labeled as dog relative to the total number of image labeled as dog, right? So that's what precision gives you. Recall is its counterpoint, and it's often referred to as sensitivity, and this is what we think of as a true positive rate. This is the number of true positives over all of the things that should have been called a positive. So this is that whole population. So in this case, it's the number of images that are accurately labeled as dog relative to the total number of dog images in the data set itself. So those are two metrics that you see a lot. There's some other ones that you'll see, and I've got them listed in this table here, but those are ones that you'll see a lot in the literature. But there are lots more. There are even more than what's on this table. There are tons of metrics that you can choose. So select your metric carefully. Accuracy is not always the best choice and is often not the correct choice for you. Do beware of misleading metrics. Metrics don't tell you the whole story. If we have a database of Gatorade drinks and we have a sample of blue Windex, our model will very confidently say that is blue Gatorade. And that's not going to help you if you're going to drink it. So be careful that misleading metrics aren't necessarily telling you the whole story, aren't really telling you, that accuracy isn't really telling you whether it's Windex or Gatorade, it's just saying it matches with blue Gatorade, right? Okay, and that last thing about misleading metrics, that pushes us straight into some of our important principles. And I know you'll go through some of these in more depth later today, but I just wanted to give you an introduction. So that first one is misleading evaluations. It's what we just talked about. You can show any metric, right? And there's lots of books that have been written about lying with statistics. There are all sorts of metrics you can select that show part of the story. So be very careful about incomplete evaluations. If you have a model that chooses true 100% of the time, it will have 100% recall. And so recall isn't enough. Precision isn't enough. You have to show kind of the full evaluation picture. So you may need more than one metric. The other part of this is that you could have misleading data. You could have data that was collected for evaluation, but that it's not realistic or it's not representative of what you're actually trying to do. If you have, for example, 99 cat images and one dog image, and your model selects cat every single time, it's going to get 99% accuracy, but that actually isn't reflective of its performance, right? Because it's just saying cat every time. You could hand it a picture of a horse, it would still call it cat. So misleading data and misleading accuracy are very important to think about and be aware of. Other important topics to consider are, and this is very, very key, it's very big, is reproducibility, especially in science and in scientific literature. Just like with a lab notebook, somebody should be able to take your data, your model, and get your same results back out. In the case of data science and machine learning, there's kind of two aspects to that that reproducibility, right? So rather than just walking through the procedure on your lab notebook, when it comes to data science and machine learning, there's the data reproducibility and the model reproducibility, and you really need both of those in order to fully have a reproducible result. For data reproducibility, there should be some documentation of where that data has come from and how it was processed or prepared for the analysis that you did. And I've got an example over on the right of what's called a data card. And this is just documentation of the trends and the statistics and the where the data comes from for a particular data set. The second piece of that is the model reproducibility. And on the bottom side of the right, I have what's called a model card. This includes all the model details that should be, that someone would need to reproduce the results that you did. That includes code, and it includes packages, so the versions of your packages and everything. It includes your training environment. So did you train it for 10 steps or did you train it for 200 steps? And finally, the actual trained model. Oftentimes what you'll see are other things like how the model was evaluated and what the model should be used for. So what the limits of its usefulness are. And those are also really good parameters to keep in mind. The last thing that I like to think about when it comes to kind of the reproducibility, but really the understandability of what you're doing and the data science you're doing is what's called explainability and interpretability. Now, this is an entire field of data science and machine learning, so we're not going to cover it all here. But overall, what you're trying to think about is how interpretable are the model's decisions to a human. If that human is taking that model's decision into court, it probably needs to be pretty interpretable. Whereas if it's just kind of one step along a workflow at a desktop, maybe it doesn't, you don't need to show the full decision path that the model took every time. You just kind of need the decision. So that ties in with how interpretable does it need to be? Do you actually have to get to the point where you understand everything, or is it okay for it to be automated and not really have that understanding all there? All right. Now, this is another big one, and this is one that I know you're covering later today, so I'm just going to kind of give you a teaser. But data science and machine learning ethics is absolutely critical. For those of you who are familiar with some of the human testing that we did in the 1950s, it wasn't necessarily ethical. And we need to be careful that we're not doing something similarly biased in the data that we collect now. So it's very important that the data, that the work that we do be responsible, equitable, traceable, reliable, and governable. Now, what that actually means for responsible is that the problem needs to be posed well and in such a way as to not be designed to harm people, right? So it has to be a responsible use of machine learning. Down in the bottom left there, you'll see a weapon and a gun. Somebody strapped AI to that. That's not your most responsible use of machine learning, and is not the goal of machine learning. So this is why thinking about the responsible part, the ethical part of your machine learning is very important. Equitable is another one that's very, very key. And when you take a more equitable approach to the data science and machine learning the goal is to minimize unintended bias. So you'll see down in the middle there, this is a bias explorer tool for some big open models that are available in the literature. And what you can do is just put in a category and it will show you images of what it thinks that category is, what that model thinks that category is defined as. If you ask some of the big models in literature to define a doctor, it will show you a male, and it will show you a roughly middle-aged, brown-haired male. If you ask for nurse, it will give you almost exclusively young women. And that is obviously highly biased and problematic if, for example, you're using this machine learning to hire your staff. And so it's incredibly important to understand the bias in your data and understand the bias in your model and work to mitigate that bias. Traceability ties back to the reproducibility. Just document, document, document. The model, the data sources, everything, same as you would a lab notebook, right? Everything should be traceable, auditable. Reliable ties into having well-defined uses, and then governable is making sure that you control the uses that you're, as best you can, that your model is being used for. So there are a few examples in the past where people have done things like release a chatbot to Twitter and let it learn from Twitter, and the internet being the internet, internet trolls come in and basically turn this chatbot into another internet troll. So, you know, as soon as you release it into the public, you kind of lose some of that. So be careful that you're thinking through how that's going to be released and where it's going to be released and how it could be used. Okay, so the very last thing is useful tools, and I'm going to provide this more as a reference for you than anything else. So we're not going to talk through this, but just so you have it, some useful tools in Python are GitHub, and then where you might be able to ask questions. And with that, I will get onto the Discord and open it to questions.
There we go. Eva, we have an in-person question.
Thank you. Hello, thank you for the great presentation. So could you give us an example of machine learning applied, non-supervised machine learning applied to multiomics?
Yeah, sort of. I don't do multiomics, so you'll have to forgive me a little. One really good example that you'll see a lot is clustering. So when you don't actually know how your data clusters, but you just want to see how things group together. A lot of times people will apply clustering. There's one diagram I have that shows gene expression between two genes and then how they split out in their clusters very nicely. And that's an unsupervised, you can do unsupervised clustering for that. I think you'll get more examples throughout the course of the week as well. So that is also a question that you can ask other folks as they talk through some of the things this week. Are there any other questions? And I'll be online and checking the Discord all day. So if you have any questions, you can drop them in there as well for me.
Tanya, did you have a question online?
Oh, I do see one, which is, do you see scientific data volume and velocity as challenges in the machine learning computational workflows? Yes. So mass spectral data can be very large and very dense, especially when you're doing like untargeted collections and things like that. And then they can also be highly complex and super noisy. And that's less an issue of velocity and more an issue of quality. Well, quality, it's high quality, it's just very dense. The other side of it is that you'll also get cases where when you do real data, if you're doing things like animal testing, you're going to get a very small number of things. So you'll have what are considered very sparse data sets. So it's not necessarily so much volume of data that you can't deal with it in traditional ways, but it's so little data that you actually have to treat it completely differently as well. So not necessarily a bottleneck, but definitely a challenge.
Hey, Eva, this is Lisa. We have one other question further up in the Discord about, could you go back to your figure about overfitting and underestimating?
Yes.
Slide with, could you maybe take one more pass at explaining that diagram?
Yes, you're gonna have to give me a second because I think I have to flip directly back. Yeah, so overfitting and underfitting is hard and challenging. So this is kind of the example that I like to use, is this kind of bottom right. It's bottom right on my screen. I'm not sure where it is on yours, where you've got the line for the model and it's got underfit on the left and overfit on the right. Underfitting is typically a case where your error is going to be very high, and it's because you're not actually predicting any of the patterns in the data at all. You draw this kind of flat line through something that's actually kind of a curve, right? So that's underfitting. Overfitting is where in training your model, you'll actually have very, very high accuracy, because what your model has done is drawn a line through every single point, but it's basically just wiggling through everything. It's fitting the noise more than it is actually fitting the pattern that you're after. So what you'll see, and this goes into the kind of figure on the left here, is that your training error, when you're actually training your model, will go very, very low. So you'll have very high accuracy, but your validation error when you play with real data after the model training will be massively high. because it's so specifically trained to the noise in the training data that it can't possibly do anything else anymore. And so that's what overfitting is. This comic on the previous slide is kind of goofy, but it's actually a very good example of overfitting. On kind of the bottom left, it's where the model fits every single point. And that's not actually a true understanding of this data at all, but you're just fitting into the noise. Does that help?
Thanks, Eva. This is Luke. One more question here. We got 1 more minute. So this is from Elisa Yang. How could we consider machine learning in microbial complex networks? For example, how do we give the biological meaning when we design the machine learning method in the construction of microbial networks, especially for supervised or unsupervised? Could you show a specific case?
So, okay, I'm not actually gonna answer this question, but I will tell you why. And the reason is that's a big piece of what explainability and interpretability is all about. So I do more than metabolomics work, right? But there is still, you need to have some kind of understanding of the chemistry when you're using these machine learning models. And the models are going from like the features to the toxicity, which, okay, cool, but what about those features are actually leading to that toxicity, to that endpoint, to that profile, whatever it happens to be. And getting back that understanding of the meaning, the chemical meaning or the biological meaning is massively complex. And it's really a big challenge. You can do things like use models that are fundamentally more interpretable in what they're doing. Things like decision trees, where you can actually walk through all the decisions that a model has made to make its prediction. But when you get into the really big, complex, making news stories, large models, it's more black box and it's harder to understand, to get biological meaning back out. There are ways to do it, but it's a very challenging problem.